Chapter 5 DNA, Dollars and Drugs. Biotechnology. Herb Boyer has a way with meetings. We have seen how his 1972 chat with Stanley Cohen in a Waikiki deli led to the experiments that made recombinant DNA a reality. In 1976, lightning struck a second time. The scene was San Francisco. The meeting was with a venture capitalist named Bob Swanson, and the result was a whole new industry that would come to be called biotechnology. Only 27 when he took the initiative and contacted Boyer, Swanson was already making a name for himself in high-stakes finance. He was looking for a new business opportunity, and with his background in science, he sensed one in the newly minted technology of recombinant DNA. Trouble was, everyone Swanson spoke to told him that he was jumping the gun. Even Stanley Cohen suggested that commercial applications were at least several years away. As for Boyer himself, he disliked distractions, especially when they involved men in suits who always look out of place in the jeans and t-shirt world of academic science. Somehow, though, Swanson cajoled him into sparing 10 minutes of his time on Friday afternoon. 10 minutes turned into several hours, and then several beers when the meeting was adjourned to nearby Churchill's Bar, where Swanson discovered he had succeeded in rousing a latent entrepreneur. It was in Derry Borough High School's 1954 yearbook that class president Boyer had first declared his ambition to become a successful businessman. The basic proposition was extraordinarily simple. Find a way to use the Cohen-Boyer technology to produce proteins that are marketable. A gene for a useful protein, say one with therapeutic value, such as human insulin, could be inserted into a bacterium, which in turn would start manufacturing the protein. Then it would just be a matter of scaling up production, from petri dishes in the laboratory to vast industrial-size vats, and harvesting the protein as it was produced. Simple in principle, but not so simple in practice. Nevertheless, Boyer and Swanson were optimistic. Each plunked down $500 to form a partnership dedicated to exploiting the new technology. In April 1976, they formed the world's first biotech company. Swanson's suggestion that they call the firm Herb Bob, a combination of their first names, was mercifully rejected by Boyer, who offered instead Genentech, short for genetic engineering technology. Insulin was an obvious commercial first target for Genentech. Diabetics require regular injections of this protein since their bodies naturally produce either too little of it, type 2 diabetes, or none at all, type 1. Before the discovery in 1921 of insulin's role in regulating blood sugar levels, type 1 diabetes was lethal. Since then, the production of insulin for use by diabetics has become a major industry. Because blood sugar levels are regulated much the same way in all mammals, it is possible to use insulin from domestic animals, mainly pigs and cows. Pig and cow insulins differ slightly from the human version. Pig insulin by 1 amino acid in the 51 amino acid protein chain, and cow insulin by 3. These differences can occasionally cause adverse effects in patients. Diabetics sometimes develop allergies to the foreign protein. The biotech way around these allergy problems would be to provide diabetics with the real McCoy, human insulin. With an estimated 8 million diabetics in the United States, insulin promised a biotech goldmine. Boyer and Swanson, however, were not alone in recognizing its potential. A group of Boyer's colleagues at the University of California, San Francisco, UCSF, as well as Wally Gilbert at Harvard, had also realized that cloning human insulin would prove both scientifically and commercially valuable. In May 1978, The stakes were raised when Gilbert and several others from the United States and Europe formed their own company, Biogen. The contrasting origins of Biogen and Genentech showed just how fast things were moving. Genentech was envisioned by a 27-year-old willing to work the phones. Biogen was put together by a consortium of seasoned venture capitalists who headhunted top scientists. Genentech was born in a San Francisco bar, Biogen in a fancy European hotel. Both companies, however, shared the same vision, and insulin was part of it. The race was on. Inducing a bacterium to produce a human protein is tricky. Particularly awkward is the presence of introns, those non-coding segments of DNA found in human genes. Since bacteria have no introns, they have no means for dealing with them. While the human cell carefully edits the messenger RNA to remove these non-coding segments, bacteria with no such capacity cannot produce a protein from a human gene. And so, if E. coli were really going to be harnessed to produce human proteins from human genes, the intron obstacle needed to be overcome first. The rival startups approached the problem in different ways. Genentech's strategy was to chemically synthesize the intron-free portions of the gene, which could then be inserted into a plasmid. They would, in effect, be cloning an artificial copy of the original gene. Well, nowadays, this cumbersome method is seldom used, but at the time, Genentech's was a smart strategy. The Asilomar biohazard meeting had occurred only a short time earlier, and genetic cloning, particularly when it involved human genes, was still viewed with great suspicion and fell under heavy regulation. However, by using an artificial copy of the gene, rather than one actually extracted from a human being, Genentech had found a loophole. The company's insulin hunt could proceed unimpeded by the new rules. Genentech's competitors followed an alternative approach, the one generally used today. But, working with DNA taken from actual human cells, they would soon find themselves stumbling into a regulatory nightmare. Their method employed one of molecular biology's most surprising discoveries to date, that the central dogma governing the flow of genetic information, the rule that DNA begets RNA, which in turn begets protein, could occasionally be violated. In the 1950s, scientists had discovered a group of viruses that contain RNA but lack DNA. HIV, the virus that causes AIDS, is a member of this group. Subsequent research showed that these viruses could nevertheless convert their RNA into DNA after inserting it into a host cell. These viruses thus defy the central dogma with their backward RNA to DNA path. The critical trick is performed by an enzyme, reverse transcriptase, that converts RNA to DNA. Its discovery in 1970 earned Howard Temin and David Baltimore the 1975 Nobel Prize in Physiology or Medicine.